Welcome everyone. Please pull out this name and DNA coding sheet from last week Wednesday. This is last Wednesday. Well, actually, this wasn't from last Wednesday. We had this, I think, the Monday before. Okay, this was even from the Monday before. We built our names in DNA on Monday as well. Okay, so pull this out, please. What we are about to do, so you went through and you coded for your name in DNA. DNA codes for a message, and that message can be coded and decoded. Your DNA model coded for your name, okay? And we should be able to go either way. We should be able to code and decode. So what we are about to do is you are about to decode. So what we will do is I'll get out a stack of DNA molecules. Everyone's, remember, was 30 long. On the bottom was a little slip of paper that had the name that the person had, okay, on the inside. When you grab one, maybe just take a peek at it to make sure it's not yours. That's really the only thing, is if you prefer that it's not yours. And then what I want you to do, somewhere on maybe just the side, somewhere on here, I want you to use this key for you to decode their name. Okay, we want you to decode their DNA and check it. Make sure they coded it right. Make sure that they didn't have a mutation. Make sure that you can decode. There have been some oopsie-doops along the way. There was one student last hour who appears to have not put they messed up their start codon. Instead of going TAC, they had CAT, which does not say start. So they had to keep scrolling, and their start started about 2 letters off, and then it was all jumbled up. They didn't have a start codon. That's called a mutation. There was one student who completely forgot a letter, so like instead of being Tom, they were T-O. They dropped an entire letter. Okay, so that was a mutation. A lot of them have been really good. Most of them have been pretty spot on. So what I'm gonna do is, while you're proofreading them, I'm gonna go through and I'm gonna be like, hey, were they good? Were they not good? If there's something that you don't think, because then I had one person do this. They wrote down like, hey, this person's name says Davies. And I'm like, how do you know that? Because they wrote Davies. I'm asking you to decode it and make sure that it spells Davies, okay? And if it doesn't, I'm gonna check with each of you, hey, does it match, does it not match? And if the answer is it doesn't, I wanna quickly have a conversation I'm gonna kind of go through with you like, okay, yeah, I agree, you are correct, this does not say Davies. Then you gotta go talk to Davies and say, hey, what happened here? Where did the mutation occur? Okay, so I want to have those quick conversations, and they're quick, they're brief, they're not painful, I promise. And then when you're done with that, you can just leave them for the time being. We're going to come back to them. We're going to do something else with them, but first we're just going to decode them. So in the back of the room, we are what, 6A? No, whatever. Right here. So you are going to grab one of those strands. Again, I would just do a quick check to make sure that you do not grab your own, okay? That you don't grab your own. Other than that, you're just gonna double check to make sure they're spelling, and then I'll come around and talk to you, and then we'll move on to the next piece. So grab a name and DNA and decode it. So if we don't have a start codon, it doesn't start coding for that message. So just like it didn't start coding for your name, or we forget to stop, it's going to keep adding random nonsense letters even though your name is stopped spelling. So mutations can happen, so we just gotta kind of be mindful of that and be aware of that. But if there's a oops, that's going to affect the outcome. Now, the last thing we're going to do with these molecules is you have 5 of these strips that are attached together. We want you to tear it off, so you just have 1. Okay, which means you have 6 base pairs hanging off the side. Okay, so somewhere, doesn't matter, tear off so you only have 6. We want something that looks like this. Okay, doesn't matter where Doesn't matter even if they have a misspelling, a mutation, doesn't matter. We're using this for something else now. Okay, so you need a total of 6 base pairs. The rest of that you can just set aside for right now. Okay, we aren't gonna use the extra parts. Okay, you only need 1 set of 6. It doesn't matter if you pick it from the middle, the top, the bottom, randomly, it doesn't matter, you need 6. Now, how many strands are in are in DNA? Is it a single strand or a double strand DNA? It's a double strand. Is this a single strand or a double strand? This is single, right? This model is incomplete. Why did I do it this way? Because one side of the DNA strand is called the coding side, the other is called the non-coding side. This is the coding side. This is the part we cared about. But I want to make sure we're clear. It is a 2-strand model. It's a double strand. And DNA runs in something that's called anti-parallel. So what you are about to do is you're going to use Chargaff's complementary base pair rule, and you are going to make the complement of the 6 that you have. You are going to put the other 6 complementary bases on over here. So, according to my model, I start with the letter G. What is the complement of G? C. So I would take the letter C, and I'm going to put it like that. What's the complement to T?
A.
And what's the complement to T?
A.
And I'm trying to line them up. Okay, and we keep going. Now, some of you are close enough that you can probably see, what did I do with my names? With the complement? They're upside down, right? I put them upside down on purpose. Now, it wasn't an accident. Because DNA runs in something called an anti-parallel. Now, when you do this, you're going to get your complements. There's this line on the red, there's this line on the blue, and assuming that they kind of put that line right on the edge, these lines over here should all pretty well match up. So when we go through and we attach our sugar phosphate backbone, they should be pretty close to the right size. Notice that my phosphate, sugar, phosphate, sugar, phosphate, sugar runs one way, but over here it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. We call this anti-parallel. These 2 strands are parallel, but they're running in opposite directions, therefore it's called anti-parallel. So when you take these 6, you are going to need 1 more sugar phosphate backbone. You only need 1 because you only have 1. You're going to have to get as many, a total of 6 base pairs. You need to figure out the complements you need. Remember, A's and T's, C's and G's. And then we want you to glue stick them together to show that anti-parallel model. The glue sticks on that paper towel are the clean, disinfected glue sticks from last hour. This piece of paper right here is meant to be that scratch paper to keep all your gluing mess off the table. And then when you're done with this, you can dispose of that in the recycling. Your used go right here. Now, if you want to right now, those extra long strands, your extra long strands can go on this stack. You want to get rid of them right now. If you want to hang on to them for a little bit, you can. So then you're going to find the complements and anti-parallel, and before you put those back, or you're done with those, I want to make sure you see them. They were, okay, perfect. So these, again, they run anti-parallel, and if I were to look at it this way, it goes phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. If I were to be looking at it standing up here looking this way, it would read the same. It would read phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. Anti-parallel. One of these is the coding side, one is the non-coding side. Now, someone asked me in my earlier hours, well, how do you know which side is the coding side? And I didn't know the answer, so I had to go and ask someone else. And then what it comes down to is there's proteins that match and like basically say start coding on this side because there's a certain sequence prior to even the start codon that say start coding here. That help identify the coding and non-coding side. It's again about proteins though. So as you are kind of wrapping those up, I saw a lot of you are pretty close, if not done already. Couple things in terms of transitioning. If you took that scratch piece of paper, that can get recycled. Glue sticks go in that bin. You're gonna want this packet, and then those 6 bases. I think I've looked at everyone now. There's a stack on that table next to. Next to those long strands. Next to the long strands of DNA, that there's a stack of double strands that you can put them on. Assuming that I looked at it, and I think I looked at just about everyone's. Okay, so there's a different stack back there for you to deposit those. Glue sticks go in the bin. There's a packet up front. Complementary bases that run in anti-parallel to each other go over on the side. So, today what we're going to start talking about is the idea of protein synthesis. Now, you had a guided notes on protein synthesis. There was an Amoeba Sister video that you should have watched, I believe that was yesterday, talk about protein synthesis. What we're going to do is protein synthesis is a process that we go through that takes DNA's coded message and eventually we assemble proteins. There's 2 steps that get us there before. From DNA to mRNA, that process is called transcription. From mRNA to tRNA, it's translation. It's all a complementary base pair. Everything follows Chargaff's rule with one exception. In RNA, that's on our Venn diagram, that was the thing, there was no T, there's a U. So everything follows Chargaff's rule with that one exception, that when you go from DNA to mRNA, instead of for every A there's a T, for every A there's a U. But then we still follow that same pair from RNA to RNA. For every U, there's a A. Okay? If we would have started with a T during transcription, for every T, there's an A. That's still Chargaff. But then for every A, there's a U, because there's no such thing as a T once we get to the RNA sequencing. Now, DNA codes for a message. That message eventually assembles amino acids, and based upon the order in which those amino acids are assembled, they make certain proteins. We're all about making proteins. Okay, proteins are what express traits. So in your Section 3 guided notes, there was an analogy about a cookie recipe. And I'll reference that again because I think it's a good one. Okay, you're at the library. The library is the nucleus. There's a copy of a recipe book that you want to make cookies from. However, the librarian won't let you take the book out of the library. Remember, the library is the nucleus. The copy of the recipe book is your DNA. The DNA never leaves the nucleus, just like that recipe book can't leave the library. Okay? But you want to assemble those ingredients of that cookie and make the cookies. And where do you make cookies? At the library or at your house? Hopefully at your house. I don't think the librarians are like that, right? So you're going to go to your house and you're going to try to make those cookies. But you need to know the ingredients, you need the order you put them in, you need to know the steps. So somehow you got to make a copy, right? Not too hard nowadays to make a copy, but you got to make a copy of that recipe book because the librarian won't let you take the recipe book, the DNA, from the library, the nucleus, home to your house. So you make a copy. That copy is mRNA. It's a copy of the recipe. It's not the copy. It's not the original. It's a copy. Okay, so you take that copy, and then that copy, in the form of mRNA, goes to your house, which is where you are going to assemble the ingredients and make the cookie. We're talking about making proteins. Anybody remember from cell parts where proteins are made? What's that? Nope, not the mitochondria. That's where cellular respiration occurs. I'll give you a clue. It gives it, some of them, a rough appearance, but it's not specifically the rough ER. What are the bumps on it that make it the rough ER? Well, it starts with an R. Ribosomes, yeah. I don't know who that was, but yeah, ribosomes. Okay, so your house is the ribosomes, and the ribosomes are where proteins are made. Your house is where you make the cookies. Okay, but we need these instructions. So that's making proteins. This is a nice ride view. Whoa! That's good cinematography right there. No one's motion's like, and that's kind of the analogy that we're going to go with here. Okay, so what this is going to kind of do twofold. It's going to go through And we're going to talk about the process called transcription and translation, and from those processes, we can code for a message. Now, eventually, what we're going to get to is we're going to start talking about assembling amino acids in a certain order to make proteins. Slightly different model. So the idea, and I'll kind of probably write this again, the same kind of information that we have written on the board, is this process that when we go from DNA to mRNA, mRNA, just for the record, stands for messenger RNA. That process is called transcription. That process is called transcription. When we go mRNA to tRNA, that process is called translation. Okay? When we go from DNA to RNA, everything is the exact same as Chargaff said, with one exception, that for every A, there's a U. Okay? That's kind of the exception here. Now, if there's a mutation, something might not work out. These should hopefully all make sense, but the idea is what we're going to do is we're going to code for a message. So we're going to make sentences. So DNA codes for a message. We coded our names with DNA. But what happens in a cell is your DNA has the original message that we make a copy to then make and assemble a sentence or a recipe, your cookies. So we're following complementary base pairing still. So for every T, there's a A. For every A, there's a T. Which would be true if we were going from DNA to DNA, absolutely. But because we're going from DNA to RNA, for every A now there's a U. Okay? For every C, there's a G. For every A, there's a U. For every A, there's a U. For every A, there's a U. For every G, there's a. For every T, there's a A. Okay, for every T, there's a A. The only thing that's different from Chargaff's rule is there's no such thing as a T, so you put a U there instead. For every A, there's a U. For every G, there's a C. And every A, there's a. Why don't you try the next 9? GAG, TAG, ATC. Complements. So GAG, what's the complement of GAG? C, U, what? C, yep. TAG, what was the first letter? A. Okay, AUC, and then ATC, the complement is UAG, right? UAG. Now, these 3 letters of mRNA are referred to as something called a codon. So I kept talking about in our name in DNA, we need a start codon. 3-letter sequence of bases is called a codon. Specifically, a codon, I don't think technically in DNA is called a codon, but in mRNA, 3 bases in a pair in a row is called a codon. The complement of a 3-letter codon is referred to as an anticodon. Okay, so in tRNA, the complement of the codon is referred to as an anticodon. Okay, it's just a term that we're going to use. So complementary base pairing, from mRNA to tRNA, it's RNA to RNA. So for every A, there's a U, right? Because there's no such thing as T's when we talk about R and A. For every U, there's a, and for every G, there's a C. For every U, there's a, for every U, there's a, for every U, there's a, for every C, there's a, for every A, there's a U. For every A, there's a U. Try the next three. We'll just double check it before I let you do the last 9. Okay, 5 seconds. What's the complement of U, C, U? All right, what's the complement of U, C, U? A, G, A. Try the last 9 if you haven't done so already. The complement of CUC is GAG. The complement of of AUC is, I almost wrote a T, but there's no such thing as a T, right? Is UAG. The complement of UAG is AUC. Hope you can't see that. Did I do that right? I hope so. Mutations happen and we want to try to eliminate mutations. Okay, now we can code for sentences. On page 3, there's a chart. This chart is the same format, layout as our name in DNA, but instead of being letters in the alphabet, it's words in a sentence now. I would encourage you to put, like right here, we put some stars next to the word start. We also put some stars right here next to the word stop. Because those aren't actually words of our sentence, those are the start and stop messages. Okay, so that's why we kind of put those there. They're in bold, but to try to really kind of make that emphasis, those are start and stop codons. Oops, not codons. Now notice how this chart, it says it's a chart for the anticodons for tRNA. Remember, anticodons are 3-letter combinations that are the complement of mRNA. This is an anticodon, and this chart codes off a tRNA. It's possible that we could code off a DNA. It's possible we code off a mRNA. It's possible we code off a tRNA. So just be mindful of what the key says. Okay? So our first sequence is UAC, and if we go UAC, the first thing in our sentence is really saying, let's start our sentence. Okay, we're going to start writing our sentence. What does AAA code for? Well, let's see. Here's an A, so that's my row. The second letter is the column of A. The third letter is the, gives us the word who. G, U, codes for the letter, or the word, I'm sorry, the word let. You try the next 4 anticodons. Who let and if there's no stop, then your sentence keeps going. Or if it says stop, you stop when it says stop. And what is our sentence? It says, who let the dogs out? Stop. Which means stop writing your sentence. We'll go through a couple of this next one together. I know some of you are kind of working ahead, and that's fine. I just want to make sure we're on the right track before we depart today. So what's the complement of TAC? For every T, there's an A. For every A, there's a U. For every C, there's a G. What's the complement of ATA? UAU. Now I'm hoping silence doesn't mean total confusion. I'm hoping that's not the case. Is anyone unclear about DNA to mRNA? Okay, complementary base pairing is how we go through. Okay, anyone else? Okay, I'll do GAA. Okay, then when we go from mRNA to tRNA, it's called let me take a step back. From DNA to mRNA is called transcription. From mRNA to tRNA is called translation. So for every codon, there's an anticodon. And what is the complement of AUG? UAC. What's the complement of UAU? AUA. What's the complement of GAA? CUU. Okay? Let's quick code the first three words together. UAC, UAC says start. AUA says what? Rock. CUU says music. Okay? Now I'm going to pause there, and if you did not get start rock music, please let me know, because that's hopefully what because before you walk out of here, I want to make sure we're all kind of on that same page. That we go from DNA to mRNA, from mRNA to tRNA, from tRNA to Word. You're going to need help with that. Okay, so then these should all hopefully make sense in terms of sentences. If all of a sudden this says like rock music beat, like rock music school, well, that maybe, I don't know. They should make sense. That's my point. If it doesn't, check it, because maybe you had a mutation. Some mutations that we've seen is like, instead of going from C to G, G to C, they went CC to G. That's called a mutation, but it affected the sentence. Okay, so keep an eye on that. This is one of your homeworks for your virtual days. The rest of this packet. DNA to mRNA, mRNA to tRNA, tRNA to sentences. There's a few follow-up questions on pages 3, I believe, and on page 4. Yep. And some of these you might need to use your section 3 guided notes to answer. Or, hey, I talked about 3 mRNAs called a codon. I talked about 3 tRNAs being an anticodon. Okay, that's the idea. Transcription, translation, those are what a lot of those questions are asking you about. Okay, so, yeah, that's one of your virtual day assignments. There's also a Quizlet test for unit, for Quizlet Stack Section 1 and Quizlet Section 3. There's 2 Quizlet tests for you to take, 2 tests, 2 screenshots, 2 different tests, 2 different screenshots, 2 different Canvas submissions. So that's your virtual day assignment for the weekend. If you have questions, please reach out to me and let me know.